Bom dia a todos. Compete-me a mim ser o moderador desta sessão, mas peço, sim, só um minuto ou dois para fazer, enfim, mostrar o meu estado de espírito em relação ao encontro de produtores, ao nono encontro de produtores de mirtilos. Como vocês sabem, ao INIAV é sempre pedido que façamos, enfim, um contributo com ideias para o programa, e já vamos na nona edição, e a questão que se punha é, os nossos produtores já sabem fazer, já sabem produzir mirtilos. Se os nossos produtores sabem produzir mirtilos, eles já têm o conhecimento da planta, já têm o conhecimento do que fazem, mas muitas vezes nós temos o conhecimento e fazemos e sabemos que resulta, mas falta-nos saber o porquê é que nós fazemos assim, o que é que está por trás disso. O meu desafio à organização, que a organização aceitou, foi, em vez de termos um especialista em extensão rural, alguém que viria aqui dizer-vos que façam assim assado, que assim resulta, e estando na nona edição, pensámos, não, vamos trazer alguém que pensa o mirtilo na sua fisiologia, no seu completo desenvolvimento. Isto quer dizer o quê? Além do como é que se faz, nós vamos perceber porque é que fazemos assim. Nós temos de saber o porquê, o que é que está por detrás disto. E não há nada melhor que ir à Universidade e pedir a um professor que sintetize o que é que nós estamos a pensar. E daí surgiu o convite à professora Rebeca Darnell, que está aqui connosco hoje, que eu não tenho problema nenhum em afirmar que será a pessoa que mais sabe sobre a fisiologia do mirtilo. Nós tivemos 2 dias de visitas técnicas, aproveito para agradecer aos produtores que nos receberam. Tivemos 2 dias de visitas técnicas com a professora Rebeca Darnell e ela muitas vezes dizia-me assim, Pedro, mas eu não sou extensionista e eu estas coisas, há aqui coisas que eu... Né? Pronto. Isto é para vos dizer o quê? Nós entendemos, mas muitas vezes não temos a visão global. E não há nada melhor do que ter alguém que pensa sobre os porquês. A professora Rebeca Darnell fez o seu doutoramento na Califórnia, na Universidade da Califórnia, em biologia. Biologia nos Estados Unidos, vocês sabem, eles têm a Plant Biology, portanto, a biologia das plantas, mas mais tarde mudou-se para a Universidade da Flórida. E durante 33 anos foi professora da Universidade da Flórida no Departamento de Horticultura e dedicou toda a sua vida a investigação e ensino à planta do mirtilo. Portanto, é impossível termos alguém com maior conhecimento que a professora Rebeca Darnell. É investigadora e professora, e, portanto, o que eu lhe pedi, e se vocês, enfim, não gostarem da apresentação dela, a culpa é minha, não é dela, porque eu é que pedi para falar sobre um tema, que é um tema, a mim, muito caro, porque eu gosto particularmente deste tema em termos de investigação, mas me parece que é importante para vocês. Vocês olham para uma planta e a planta tem, e nós em fisiologia falamos sempre nos pontos de produção e nos pontos de consumo. Perdoem-me aqui esta tradução livre de sourcing, mas pronto, os pontos em que se produzem os carboidratos, vocês sabem que as plantas, e a Rebeca vai explicar isso tudo, que as plantas utilizam são os produtos da fotossíntese. E, portanto, estes produtos da fotossíntese são produzidos e estão localizados em zonas diferentes. E zonas diferentes das plantas vão ter comportamentos diferentes. Umas vezes produzem, outras vezes consomem. E, portanto, nós temos... É este balanço que ela vai falar. Espero que seja proveitoso para vocês todos. E o que é que vamos fazer? A apresentação é em inglês. Eu, normalmente, acho que devemos assumir que deixar fluir uma apresentação, mas a organização foi muito severa comigo e disse, não, vamos ter que parar porque vai ver se as pessoas estão a entender o que se está a passar. Portanto, nós vamos ter alguns momentos em que paramos, eu vou tentar resumir minimamente mal, como é evidente, mas pronto, perdoem-me aqueles que vão entender tudo, os que não entenderem tudo, depois não se vão preocupar com isso, não é? Mas pronto, mas eu vou tentar só resumir o que ela disse em português, para que haja também aqui um certo equilíbrio entre a apresentação. Mas eu pedi à doutora Rebeca Darnell que falasse devagarinho. O inglês é relativamente simples. Os termos são novos. Portanto, o único 2 termos que vocês têm que entender muito bem é que ela vai falar sempre sobre a relação entre os locais de produção ou as fontes de carboidratos e onde eles estão a ser utilizados em cada momento do ciclo da planta, ok? Isto é muito importante para vocês, enfim, terem a compreensão da apresentação. Não vou alongar mais. Professor, it's your time to come. Okay? Thank you.
I have no idea what Pedro just said, so no idea at all. So first, I really want to thank the Portugal blueberry growers and Pedro for being such a gracious host and for inviting me here to give this talk. It's a real honor for me to be here, and so thank you all. Thank you all for attending, all the growers. And I think, as Pedro said, I'm going to be talking about source-sink relations in blueberry. And so really what I'm going to be talking about is photoassimilates and the end product of photoassimilates, which are carbohydrates, and how the plant moves those carbohydrates to different parts as the different parts of the plant compete with each other for the available carbohydrate. So here we have just a cartoon of a plant. So we've got no slide. There, okay, I was looking for the pointer. There it is. So here we've got leaves that are photosynthesizing, so they're capturing CO2 from the air, and ultimately they're converting that into carbohydrate in the form of sugar. When we talk about source sink, it's jargon that we use in plant science, and so the definition of a source is simply an organ that supplies carbohydrate to other parts of the plant. A sink, on the other hand, is an organ that may be able to make some of its own carbohydrate, but not enough to maintain itself. And so it must import carbohydrate from sources. So what are some common sources that we think about? Well, the mature leaves are the obvious major source on any plant, and that would include blueberry. So this is where photosynthesis is occurring. This is where that photosynthate is ultimately being changed into sugars that are being translocated to the rest of the plant. The other sources on a blueberry, though, include, at certain times of the year, perennial roots and canes, as well as the crown. And so these organs on a blueberry plant change from sources to sinks throughout the season, depending upon the stage of development. So at some time during the season, they can act as sources of carbohydrate to parts of the plant that need it. And typically that time is when the leaves are no longer able to photosynthesize, and in a deciduous system, that would be in the wintertime. Our sinks are going to be flowers, fruit, the shoot and root apices, in other words, the growing points of both the shoot and the root, and then these same perennial parts, the roots and the canes, at other times of the year, times when there are leaves present on the plant that are actively photosynthesizing. As I've said, sugars are ultimately the end product of photosynthesis, and sucrose, which is just common table sugar, is the type of sugar that the blueberry transports throughout the entire plant in order to get carbohydrate to the leaves, to the flowers, to the fruits, to the roots, to the crown, wherever it's going to be. That sucrose is transported in part of the vascular system of the plant called the phloem. And so the phloem is a continuous series of cells that connect all parts of the plant, the roots, the crown, the stems, the leaves, the flowers, the fruits. They are all connected with phloem, and that's how the sucrose is being moved around. So here's just a cartoon again that shows you how that's happening, I hope. So here we've got just another cartoon of a plant. Here we've got the two parts of the vascular system, the xylem, which is what is transporting water and nutrients in the soil up, always up, into the upper parts of the plant. And then we've got the phloem in pink that again is transporting photoassimilates. And so here what you see is photosynthesis is occurring in this leaf, that sucrose is being moved into the phloem, the pink, and it can be translocated up into a growth sink at the top, let's say an expanding leaf or a flower or a fruit, or it can be moved down to another sink. It could be a flower or a fruit that's below that leaf, or it could be going down into the roots where it's going to be supply the carbohydrate required for root growth, or it could be stored in those roots in the form of starch, which is just a long carbohydrate. It could be stored in those roots or in the canes as starch and then held there in reserve for the plant to access at times when there isn't enough leaf photosynthesis for its needs. So what happens then in a deciduous system is, as we get into late fall right now, the leaves will begin to upsize. They'll fall off, and now there's no leaves for photosynthesis. So we have no current source of carbohydrates for the plant, and so that's when these stored reserves in the form of starch come into play. So that starch is broken down into sucrose. That's our sugar that's being translocated throughout the plant. And then that sucrose is being moved into the phloem, and it is going to be moved into the parts of the plant that need it even during the winter, which are primarily the flower buds and the vegetative buds. They're alive, they're metabolizing very slowly, but they are developing, and so they need carbohydrate to do that. And then the rest of the tissue that just needs carbohydrate in order to respire. Regardless of where the sucrose is coming from, whether it's coming from current photosynthesis or whether it's coming from reserves that have been stored and then remade into sucrose, it's going to be sent to two types of sinks on a plant, and blueberry is no exception. So we have metabolism sinks, also referred to as growth sinks. So these are the ones that you can see. And so these are the expanding buds in the spring, the expanding floral buds and vegetative buds. They're the growing shoot apices, the root apices, and the very young leaves. That contrasts with the second type of sink that I've already mentioned, and that is a storage sink. So once again, these perennial parts of the blueberry, the canes and the roots and the crown, can act as sinks, typically during the summer when the leaves are still on the plant, and then they will act as sources of carbohydrate when there are no leaves present on the plant. The thing to keep in mind is that all of these sinks, the flowers, the fruits, the young leaves, the shoot apices, the root apices, all of these sinks are competing with each other to get this available carbohydrate. There's only so much carbohydrate available to go around, and so some of these sinks are going to win, and some of these sinks are going to lose. Here's just a picture of a non-reproductive blueberry plant. So our primary source is going to be the mature leaves. That's where the sucrose is being made and translocated to the rest of the plant. So again, it could be translocated up into the shoot apices, where it's going to supply the carbohydrate to those very, very young leaves that are just beginning to grow. And it's going to be transported down into the crowns and the roots. And when it goes down into the crowns and the roots, it's going to supply the carbohydrate to promote new root growth, those white roots that we want in order to take up nutrients and water. And it's also going to be stored in the older roots. These thicker roots are going to become storage sites for that carbohydrate, again in the form of starch. And those roots are going to hold on to that starch until it's needed later in the season. Sources and sinks, and I've already said this, can change during the course of the year. And so if we start up here at the upper left, here we have a blueberry plant, a lot of developing fruit on it. In this case, the primary sink is the fruit. But we also have new vegetative growth occurring. And so vegetative growth in blueberry, the shoot growth occurs episodically. So we get shoot growth and then it stops. We get shoot growth, stops, shoot growth, stops. All of that shoot growth is a sink for the carbohydrate. So it is competing with those fruit for the available carbohydrate that is coming at this point from the primary source, which is the leaves. After the fruit is harvested, now we move over to the post-harvest situation where we've just got the plant growing until for the remainder of the season. So now the primary sink is vegetative growth, both shoot growth and root growth. And at this point, as we get closer and closer towards the end of summer and early fall, we're beginning to develop and differentiate flower buds in the nodes of the leaves as we move down the shoot from the top. So we begin to develop flower buds in each of those nodes down to a certain point depending upon environment and the genetics of the plant. At this point then, those are the primary sinks, the vegetative growth and the development of the flower buds. The primary source is still the leaves because we've still got lots of good leaves on those plants that are photosynthesizing. As we move into late fall and early winter, now these leaves are getting ready to upsize. And so the primary sink, again, is the development of the flower buds. And it's also at this point storage carbohydrate because it's prior to this point, so between this point and this point, that some of that carbohydrate in response to environmental conditions is being moved back into the plant and down into the crowns, the roots, and the canes so it can be stored and used later by the plant. And so in this situation, we've got two sinks, flower buds and storage reserves, and again, the primary source is leaves. Once these leaves upsize, now we move down into a situation where the plant is dormant, hibernating. Now the primary sink is just basal metabolism. The plant is not visibly growing, but it's alive, it's respiring, and it must have carbohydrate to support that respiration rate. And so we refer to that as basal metabolism. And the primary source are the storage reserves that we made up here, because there are no leaves on the plant that are there to make more carbohydrate. There's no photosynthesis going on. And so the plant must rely on these reserves that it's stored prior to leaf abscission. As we move into early spring, now we're beginning to get bud swell, both flower buds and vegetative buds. Those also are very strong sinks, and they need carbohydrate imported to them to support that growth that they're beginning. So the primary sink are both the vegetative and floral buds. The primary source at this point, there are no leaves, so the primary source is still the storage that we accumulated the previous season. And so I think you can see that storage carbohydrates are really critical to good plant development and good yield. As we move into bloom, here we've got open flowers, but we also have vegetative buds that are breaking, so those again are our two primary sources, or sinks, our two primary sinks. And our sources at this point is still storage because we don't have a leaf canopy established for photosynthesis. After the leaves become more established and the fruit begin to develop, then we move back up into where we started, where we begin to get rapid fruit development. We have a vegetative canopy established, and now we don't use storage reserves anymore. Now, in order to support the sink demand of the flowers, the young developing fruits, and the leaves, now the primary source is photosynthesis from the leaves. Stop.
Thank you. Bom, vamos parar aqui um bocadinho porque, como vocês estão a ver, estas são todas as partes de definição do que nós vamos falar a seguir. Esta é a parte, isto tem que ser entendido, tem que estar entendido que nós temos um local que fornece os fotoassimilados e temos um local que vai receber esses fotoassimilados. E a ideia importante que vai ser desenvolvida agora a seguir é o quê? Estes fotoassimilados são escassos, vamos explicar porquê, a Rebeca vai explicar porquê. Estes fotoassimilados de mirtilo não chegam para tudo, ok? Portanto, os fotoassimilados, isto é a ideia básica disto tudo, não vão chegar para tudo. A nossa planta de mirtilo não vai ter fotoassimilados para tudo. Portanto, nós temos competição de vários órgãos da planta que vão competir por esses fotoassimilados. Eu pedi à organização que me deixasse ficar o slide, o último diapositivo da Rebeca, exatamente. Pronto, aqui o que é explicado? Estamos a explicar então em que momentos é que estes movimentos, um órgão que fornece fotoassimilados pode de repente ser ele próprio o consumidor dos fotoassimilados. E outra noção muito importante é a força com que isto é feito. Ela vai explicar a seguir. Portanto, é assim, há uma competição dentro da planta, a planta compete dentro dela própria, e isto do mirtilo é extremamente importante. Como eu costumo dizer, o mirtilo autorregula-se mal, e portanto, sendo uma planta que se autorregula mal, se nós deixarmos e a Rebeca vai explicar, se nós deixarmos haver um consumo exagerado por parte de um determinado órgão, nós podemos estar a comprometer os outros. Levantando aqui um bocadinho o véu daquilo que ela vai dizer, há aqui duas fases que vão ser fundamentais. E a fundamental, nós vamos ter que intervir, ela vai explicar, quando nós alteramos o que fazemos, e estamos a falar de podas, de mondas, o que nós vamos poder alterar a nossa planta de mirtilo. Portanto, fundamentalmente perceber até aqui, nós temos locais de produção e consumo, eles variam ao longo do tempo, eles têm uns mais força do que outros e no ciclo vegetativo da planta este entendimento tem que estar feito, se nós vamos cometer erros, porque estamos a fazer intervenções que nos vão prejudicar neste balanço, ok? Pronto, espero que... Ok? So, Rebecca.
Regardless of the whole source sink situation, the source supply, the carbohydrate, either from current photosynthesis or from reserves, must be greater than or equal to the sink demand. Otherwise, sink growth is going to be limited. And I'll give you the bottom line now. It appears in blueberry, sink growth, particularly fruit yield, is always source limited. And so this is the cycle for blueberry development during the course of the year. We start with, right here, bloom and leaf bud break. So this indicates what I'm going to call the first period of direct competition between two competing sinks. In this case, the flower buds and the leaf buds. We then move into fruit development and shoot growth, right here. This also represents a time of direct competition. Now we're talking about fairly rapid expansion of the fruit, and in some cases, rapid, although episodic, shoot growth. The plant then, we harvest the fruit, right here, and then we move into the third period of direct competition, and that's between the development of the flower buds and the ability of the blueberry plant to accumulate reserves. So those two things are in direct competition with each other. We then move to dormancy, the leaves are lost, and then we end up back the following spring with the competition between bloom and leaf bud break. So if we start first looking at the competition between bloom and leaf bud break, I'm going to show you some data slides from each of these three periods of limitation or competition. First, I want to tell you or remind you of how a blueberry fruit grows. So this represents days after anthesis. This is fruit dry weight. There are three stages to blueberry fruit growth. Stage 1, right here, is primarily a little bit of cell division early on, and then some cell elongation. Stage 2 is when the seeds develop. And so it's not a big sink. The sink demand during stage 2 is not very great. Stage 3, we get very rapid fruit enlargement. A very, very strong sink demand at that point. And so the two major periods of sink demand for a blueberry fruit is stage 1, in order to support the cell division that's going on and the little bit of cell enlargement. And then stage 3, where we have very rapid fruit growth. Those are very, very strong sink demands periods for a fruit. These are data looking at the use of carbohydrate reserves to support stage 1 of blueberry fruit development, when there's some cell division and a little bit of cell expansion going on, very, very early after bloom. And so what we have here are just 2 years of data, 2016, 2017. This is looking at carbohydrate reserves in roots and canes. This is in northern highbush blueberry. And so these are the stages of development from dormancy to bud swell, shoot expansion, green fruit, ripe fruit, and then this is after the fruit has been harvested and the plant is just growing vegetatively. So the important thing to note here is that in both years, we get a rapid decrease in the carbohydrate reserves in the roots and to a limited extent in the canes, but the roots, the primary reserve source. And you can see that's true in both 2016 and 2017, this rapid decline. And so it's those cane and root reserves that are supplying the carbohydrate required to support bloom and early, very early fruit development, as well as leaf bud break. Following fruit harvest, you can see a rapid increase in the carbohydrate reserve accumulation in the canes and the roots, because these are going to be required for the 2017 season. So these carbohydrates here came from these pools of carbohydrate that the plant accumulated the previous season. This is work that we've done looking at southern highbush. The pattern is exactly the same. And so again, we've got starch. Remember, that's the storage form of carbohydrate. We've got it in the roots and the canes. Again, the roots are the primary sources for the carbohydrate reserves. And as we go from dormancy to bloom, and this represents fruit development, early, mid, and late fruit development, you can see that the carbohydrate reserves are being used up until very early fruit development. And then we begin to see a slight increase as the carbohydrate reserves begin to increase again. And so what this is showing you is simply that carbohydrate reserves are supporting the sink demand of the fruit during stage 1 of fruit development. That's not the only thing that's supplying carbohydrate to the fruit, however. The other thing that is supplying carbohydrate to the fruit during this stage 1 of development is fruit photosynthesis. Blueberry fruit have an extraordinarily high photosynthetic rate compared to any other green fruit. As long as the fruit is green, it will photosynthesize, and the work that we have done suggests, depending on the cultivar, and the work we've done is all with southern highbush, depending on the cultivar, fruit photosynthesis can supply up to 25% of the total carbohydrate required to develop that fruit to maturity. So we're talking about the carbohydrate required for dry weight of that fruit and the carbohydrate required for the respiratory loss. That's a huge amount of carbohydrate that's being supplied by fruit photosynthesis. This is unheard of in any other crop. Furthermore, during the first 10 days after anthesis, about 7 to 10 days, again, cultivar specific, about the first 7 to 10 days after bloom, 80% of the carbohydrate that fruit needs is coming from itself. It is photosynthesizing enough to supply 80% of what it needs for the first 7 to 10 days after bloom. And so what that means is that during that very, very early period in stage 1, it's not a particularly strong demander for carbohydrate from other parts of the plant. Excuse me, in other words, the reserves, because it's supplying so much of its own. Leaf bud break, however, is a sink. However, new leaves will become a source, they will become net exporters within 7 to 10 days of bud break. And so they don't have to be very large to reach the point where they are photosynthesizing enough for themselves and photosynthesizing enough to send to other parts of the plant. It only takes, at age 7 to 10, those leaves are already exporting carbohydrate, which means that your typical northern highbush blueberry has an advantage in terms of source supply during stage 1 of fruit development. And that advantage is that most northern highbush blueberries break vegetative and floral bud at about the same time. In some cases, vegetative bud break occurs a little bit before floral bud break. Since the flowers, the very young fruit, are almost self-sustaining the first 7 days or so after bloom, so they're not demanding a lot of carbohydrate from the reserve pool, that means that there's a temporal separation in the sink demand between the flowers, the young developing fruit, and the leaves. And so they're not directly competing with each other during that first 10 days. If you contrast that with your typical southern highbush, they tend to break flower bud before vegetative bud. What that means is that those very young developing fruit, although they also are self-sustaining for about 7 to 10 days in southern highbush, after that point, they change to being very strong demanders of carbohydrate. That occurs at the same time that the leaves are strong demanders of carbohydrate, which means we've set up a head-to-head competition between the young developing fruit and all of the leaf bud break in a southern highbush blueberry. They are both competing directly at the same time for the reserve carbohydrate that's available. So that is a disadvantage for southern highbush blueberry.
Vamos respirar bocadinho. Espero que esteja, enfim, a ser relativamente fácil o entendimento do que a Rebeca está a dizer, e vou só, ressalvar aqui 1 ou 2 pontos. Portanto, o que é que ela disse? Há 3 momentos, há 3 fases importantes de competição pelos fotoassimilados. Portanto, o mirtilo, mais uma vez eu digo, o mirtilo os fotoassimilados não chegam para tudo, e o que ela está a chamar a atenção é que há 3 momentos em que nós estamos em competição, ou seja, há competição por esses fotoassimilados que não chegam para tudo, ok? Esta ideia de que nós estamos sempre numa planta que não consegue produzir tudo o que precisa é importantíssima, ok? Por isso é que há competição. Há aqui uma altura em que nós entramos em competição. Pronto. Portanto, são 3 fases. Ela está a explicar a primeira fase. A questão que temos em relação a isto. Perceberam que, em relação ao desenvolvimento do fruto, há 3 momentos de desenvolvimento do fruto. Há um momento que exige reservas, há um momento que é quando nós já temos o fruto vingado e ele anda ali, nunca mais se desenvolve, não é? Pronto. Enfim, está essa fase não consome muito, mas depois quando o fruto começa a ter o seu maior desenvolvimento, então aí sim ele é enfim, absorve muito dos fotoassimilados. É evidente que nós nesta fase é fácil de entender. Tudo vem das reservas, não é? Nós não temos ainda nada, portanto a nossa planta está a sair da dormência, portanto nós estamos a ter que utilizar tudo o que nós deixámos lá do ano anterior, não é? Pronto. Portanto, o mirtilo está a ir buscar tudo às reservas, não tem qualquer dúvida. Interessante, ela se chamou a atenção, o mirtilo é uma planta bastante especial, vocês trabalham com uma planta bastante, enfim, tem assim umas manias, como eu costumo dizer, e uma das manias que tem é que consegue fazer fotossíntese através dos frutos. Isto, ela esteve a chamar a atenção, que não é tão, enfim, residual quanto a gente possa pensar. Okay? Portanto, os nossos frutos verdes, não é? No estado são importantes, e eles quantificaram, e não é, há uma determinada altura, 80% dos hidratos de carbono que o próprio fruto precisa, ele produz, não é? Só vai buscar um bocadinho, não é? E esse contributo é um contributo muito forte. Portanto, a fotossíntese do fruto é muito importante. E depois é este diapositivo que aqui está. E isto é muito importante para os produtores, quando nós estamos a trabalhar com mirtilos do Norte, não é a mesma coisa que estamos a trabalhar com mirtilos do Sul. ok? Em termos de competição pelos fotoassimilados. O que acontece, ela explicou, portanto, os mirtilos, os saudaneibos, portanto, os mirtilos, nós chamamos do tipo sul, portanto, os mirtilos do tipo sul, o que acontece? A planta vai precisar, nós temos dois pontos da planta que estão a consumir ativamente os nossos recursos, que são as flores e que são e os frutos, e depois nós vamos ter, enquanto que nós nos mirtilos do norte já vamos ter folhas, ela vai explicar que a capacidade fotossintética da folha do mirtilo, mais uma característica do mirtilo, é extremamente rápida, o que é bom para nós, não é? Mas pronto, nós nos mirtilos do sul temos que tomar atenção, porque estamos a depender 100% das reservas, enquanto nos mirtilos do norte não estamos, ok? Pronto. The floor is yours.
Thank you. So that's the first period of direct competition. Potential source limitation is between bloom and leaf bud break, and they're competing for reserve carbohydrate. Once that we move through stage 1 of fruit development, now the source change. The source changes from the reserve carbohydrates, as we've already talked about, to the new leaves that are being established on the plant. And so again, these blueberry leaves become net exporters of carbohydrate by 7 to 10 days after that bud is broken. And so very quickly they establish the vegetative canopy, and then current photosynthesis becomes the primary source for the rest of blueberry development. So fruit development and shoot growth are the second period of competition. So you've seen this slide already. This is just looking at, in southern highbush, the use of carbohydrate reserves during stage 1. And what we know is that after stage 1, or towards the end of stage 1, now current photosynthate is responsible for supplying all of the carbohydrate to those developing fruit. At the same time, they're responsible for supplying carbohydrate to the shoot apices and the root apices. So again, we've got competition. We believe that photosynthesis in blueberry is limiting. And so this is some work looking at photosynthetic rate in blueberry versus light intensity. Light intensity of full sun is about 2,000 ppf. So what this shows you is two pretty important things, I think. And that's, first of all, that single leaf blueberry photosynthesis saturates out at about 25% of full sun. If you increase full sun above that, you do not get an increase in photosynthetic rate. It plateaus out. The second important thing is that photosynthetic rates are relatively low in blueberry. A typical strawberry plant has 13 to 15. Blueberry, 5 to 7. Again, it's genetic. I've measured photosynthetic rates of 10, but typically it's not that high. So blueberries have low photosynthetic rates, and the photosynthesis saturates out at relatively low light intensities. What that means is that we think photosynthesis is limiting in blueberry in terms of fruit yield. This is some work that was done where they labeled with a heavy isotope, they labeled a leaf with CO2, and then they followed the carbohydrate to see where it went in the blueberry plant. This was in northern highbush. What they found was that the fruit got some of it, the root didn't get very much, the current vegetative growth got quite a bit, and the wood got a little bit, the canes. And this was during stage 1 of fruit development, towards the end of stage 1, after the leaves had established. When they looked at stage 3 of fruit development, they did the very same thing. Here's the leaf that they labeled with the CO2. This is where the carbohydrate ended up. Again, you can see most of the carbohydrate ended up in the fruit, but the current shoots got just as much carbohydrate as the fruit did. And again, the roots and the wood did not. So what this is showing you is that direct competition between the fruit and the vegetative growth for current photosynthate. We did some work in this was all northern highbush. We did some work in a couple cultivars of southern highbush where we had a control plant that we just let flower and fruit and photosynthesize normally. And then we had one where at the beginning of stage 3 of fruit development, we removed 80% of that fruit. When we did that, we got a huge flush of vegetative growth. And so by the end of the season, you can see the dramatic increase in leaf dry weight in the plant that we removed 80% of the fruit. Again, just support for the idea that fruit growth in stage 3 and vegetative growth in the blueberry plant are in direct competition with each other, and that there's a source limitation. If there was not a source limitation, we would not see this increase in leaf dry weight in the 80% fruit removal treatment. So what we know is that both fruit growth and vegetative growth compete with each other, and this is during stage 3 of fruit development, and that there's very clearly a source limitation, that is a current photosynthesis limitation to fruit during stage 3, which is the cell enlargement stage.
Bom, estamos já na segunda fase, enfim, deste uso dos recursos da nossa planta. Parece-me que aqui é bastante, enfim, fácil de perceber. O que aconteceu? Mudámos completamente de paradigma, não é? Pronto. Neste momento, a origem dos nossos carboidratos passam a ser da fotossíntese, ok? Já temos folhas. Portanto, a partir de agora, nós já não vamos depender das reservas da planta, nós já temos um órgão que está a fornecer os fotoassimilados. Dito isto, o que é que a Rebeca explicou? Portanto, é fácil de ver naquele diagrama que quando nós começamos a ter fotossíntese, ainda estamos numa fase de fruto, mas já começa a haver, o próprio sistema radicular começa já a receber reservas. Isto é porquê? Porque, pronto, já não está a utilizá-las e já está a subir esse nível. Outra coisa extremamente importante, e isto é outra especificidade do mirtilo, o mirtilo tem duas características muito importantes em relação à sua fotossíntese. Isto é ali para o Manel, é isto que você vai ter que ver. Nós vamos ver isso na nossa situação. A questão é assim, está lá, ele está ali, portanto, o que acontece? O mirtilo satura, portanto, a sua capacidade fotossintética satura muito rapidamente. Nós estamos com o que ela chamou 25% da intensidade luminosa normal do ar livre, portanto, que nós temos o que nós chamamos em pleno sol. Em pleno sol, fica saturada, o mirtilo não consegue produzir mais fotoassimilados, ok? Isto é extremamente importante porque é muito baixo, relacionado com outras fruteiras, né? Pronto. Por exemplo, vamos dar um exemplo, nós na framboesa temos a saturação aos 1200 micromoles e aqui nós temos aos 500, portanto há aqui um jogo muito baixo. E depois esse aspecto é fundamental, muito baixa intensidade luminosa e também a quantidade de fotoassimilados produzidos. Portanto, trabalha muito abaixo e produz muito pouco. Okay? Portanto, nós naquele eixo temos a produção fotossintética, e a produção fotossintética do mirtilo é extremamente baixa. É 1/3 do morangueiro, é metade da framboesa. Okay? Portanto, a nossa folha produz num nível de radiação muito baixo e está a produzir muito pouco. Okay? Portanto, é uma folha um pouco eficiente. Por isso, nós temos um problema de ser uma planta que está limitada pela fotossíntese. Okay? Muito importante. Depois ela mostra aqui que eles marcaram, o carbono marcado é um carbono mais pesado, portanto, que nós podemos depois ir ver onde é que ele ficou. Nós fornecemos o CO2 marcado e depois a seguir vamos ver onde é que ele está. Eles foram ver onde é que estava e o que é que acontece? Acontece que quem é que está a competir então por esse carbono? Quem está a competir por esse carbono, importante, são os frutos e o crescimento vegetativo. Portanto, um momento muito crítico que nós temos a planta está a tentar crescer, a planta está a tentar produzir e há aqui uma competição enorme, ok? Pronto. Portanto, é isto que eu tenho que chamar a atenção. Muito obrigado.
So that's the second stage or second period of direct competition between the fruit development and shoot growth, and in this case they are competing for current photosynthate. Following fruit harvest, then, we have a third period of competition, And that's when carbohydrates are required to initiate and differentiate flower buds in the nodes, and also the carbohydrate is required to start building up the reserves in the roots and the canes that will serve as carbohydrate sources during the winter and into the spring. And so now we're talking about this competition, flower bud initiation and reserve accumulation. Flower bud initiation in blueberry is environmentally controlled. It depends on short days and cool temperatures. And so here we've got some work we did with southern high-bush blueberry. We looked at an 8-hour day, and then we looked at an 8-hour day with a 1-hour non-photosynthetic night interrupt. And so what we looked at was the amount of flower bud initiation that occurred, and so all this shows you is that yes, short days are required for flower bud initiation in blueberry. And if you interrupt that short day, you don't, in this case we interrupt the night, but it's really the long night that's required, that you wipe out flower bud initiation. The other thing is it's temperature dependent. And so if we do this whole thing at 28 degrees, we wipe out flower bud initiation. So the environmental factors that are required for flower bud initiation in blueberry are short days and cool temperatures. And unfortunately, those are and this is northern highbush, same thing. This is work from Bernadine Strick's lab, where they don't get any flower bud initiation under a 16-hour day length, and they get quite a bit under an 8-hour day length. The problem is that those cool temperatures and short days are the same environmental factors that trigger reserve accumulation in blueberry. And so this is some work that we did looking at flower buds. This is flower bud density versus how much reserves were accumulated in total in the canes and the roots. And what we see is that as the number of flower buds increases, the amount of reserves decrease. In other words, the flower bud initiation and differentiation process is getting more of the carbohydrate. It is a stronger sink, and that's decreasing the amount of carbohydrate that's going into the reserves that will serve the plant once the leaves are gone.
Aqui o que é importante chamar-vos a atenção é, nós agora estamos já vamos para a terceira fase, e o que acontece? Precisamos dos dias curtos, portanto, a redução do fotoperíodo e da temperatura para começar a haver a acumulação de reservas. Mas, o que ela está a chamar aqui a atenção, cuidado, que nós estamos também a ter a diferenciação floral. E a diferenciação floral compete com a acumulação de reservas. E é este último slide que está aqui a mostrar, não é? À medida que nós temos mais botões florais florais no outono, nós estamos a competir com a quantidade de reservas que estamos lá em baixo. Portanto, isto está a preparar, vocês já estão a ver, que isto depois vai dar tudo, não é? Nós, quando intervimos na planta do mirtilo, temos que ter tudo isto em atenção, ok? Tudo isto vão ser motivos para nós termos em atenção quando é que nós vamos intervir. Nós normalmente intervimos no mirtilo depois quando vamos fazer a poda, não é? Pronto, atenção, acumulação de reservas, a diferenciação floral interfere na acumulação de reservas, ok?
So the third period of direct competition is between flower bud initiation and reserve accumulation, and that competition is for current photosynthase. And so now we have 3 periods that are pretty well defined where we have potential source limitation to blueberry fruit yield. The first one is the reserve carbohydrate limitation during stage 1 of fruit development, cell division, where we have bloom and very early fruit development occurring at the same time that we have leaf bud break. The second period of direct competition is between the fruit enlargement during stage 3 and continued vegetative growth, both shoot growth and root growth. The third period of direct competition that could limit yield is after fruit has been harvested, when you've got flower bud initiation occurring and reserve accumulation occurring at the same time. Now remember, these 2 periods are dependent upon current photosynthesis. This period is dependent upon reserve carbohydrates. So of these 3 periods, which is the most detrimental in terms of blueberry fruit yield? We've asked that question, and we've looked at we've done some work, and we believe that it is the reserve storage that has the greatest negative impact on blueberry yield. That it's the ability of blueberries to accumulate a lot of reserves that would lead to improvements in yield compared to the other two periods of source limitation. And the reason we believe that is this work where we did with a couple of different cultivars where, again, we had a control, we didn't do anything to it. We had a treatment where we removed 80% of the flower buds the fall prior to bloom in the spring. And then we had the treatment, we've already seen this treatment, where we removed 80% of the fruit at the beginning of stage 3. So what we were trying to do was differentiate these periods of potential source limitation. So here we're looking at source limitation during stage 1, that very early, that's competing for reserves. And here we're looking at source limitation during stage 3, where they're competing for current photosynthate. What we found was that, obviously compared to the control, we had reduced fruit number in both of those treatments. But what surprised us somewhat was that when we removed 80% of the flower buds, we had many, many more fruits than if we removed 80% of the fruits at the beginning of stage 3 of fruit development. And so what likely happened in this case is, although we removed 80% of the flower buds, the fact that we have many more fruit suggests that there was just better fruit set for the flower buds that remained. The other thing that surprised us was that we got greater fruit, and this is fruit dry weight, not fresh weight. We got greater fruit dry weight in the flower bud removal treatment. And so not only compared to the fruit removal treatment, not only did we get more fruit, but we got larger fruit, which we did not expect at all. We typically think you have more fruit, they're smaller. In this case, we've got more fruit and they were larger. So that led us to conclude that it was this usage of reserves that was the major constraint to blueberry yield. And that if we could increase the carbohydrate reserves, which we did when we did this treatment, that the remaining flowers, we would get good fruit set and good fruit size, and that's exactly what happened. The reason that happened, we believe, is that 80% of the cells in the blueberry fruit are formed before that flower even opens. So when that flower is still in the bud stage, 80% of the cells are already formed. And furthermore, we know that not only for blueberry, but for all fruits, that fruit size is more dependent upon cell number than it is on cell size. And so doing a treatment where we can increase cell number, like thinning at the flower bud stage, has a much bigger impact on the remaining fruit than doing a treatment where we're thinning at the cell enlargement stage. So that's exactly what we see here.
Parar aqui porque acho que isto é extremamente importante e não é tão claro quanto isto, né? Pronto. Porque mesmo para a Rebeca, que ela deve explicar, né, eles ficaram muito confusos com estes resultados. A questão aqui é, normalmente os produtores, quando têm uma carga a mais, dizem, eu agora reduzo aqui um bocadinho do frutos, tiro um bocado de frutos e esta coisa compensa. Se não compensa nada, o estrago já está feito. Okay? Pronto. E o que ela está a dizer é que mais do que isso, não é a remoção das flores após o abrolhamento, é a remoção das flores antes do abrolhamento. Ok? Porquê? Quando lá vamos nós outra vez ao mirtilo, que é uma planta muito estranha, o mirtilo o que é que faz? Aquela primeira fase que nós falámos do fruto, primeira fase de fruto, okay? Aquele aumento do número de células dá-se na estação anterior. E como o aumento do número de células é dado na estação anterior, o que acontece? Nós temos que reduzir o número de flores na estação anterior, não durante a estação. Ok? Portanto, isto agora aqui é um bocadinho confuso, mas é os resultados deles. Ok?
So are there ways we can help alleviate this problem? And yes, there are within limits. And so what we're really interested in doing is making sure that we get sufficient reserve accumulation occurring during the growing season. Because remember, those reserves are coming from current photosynthesis. And then they're going to be used to support what we believe is the most critical competition period for blueberry fruit. So obviously maintain healthy leaves after fruit harvest. That's a given. So you have to keep the leaves on as long as possible because that's going to put more carbohydrate into your reserve pools. Also, you want to prune, and I've seen some growers that have also trained their plants so that they can maximize the leaf surface area that is exposed to light. In other words, you want to increase light penetration into the canopy. So we saw this already. This is the light response curve for a single leaf, and again, it saturates out at a very low light intensity. But that does not translate into whole canopy photosynthesis. And so when you look at whole canopy photosynthesis, what you find is that, yes, although the individual leaves are saturating out at 500 ppf, at 25% of full sun, the whole canopy photosynthetic rate continues to increase all the way up to full sun, because even though these outer leaves are at this plateau region, the inner leaves are not. They're still light limited. So as long as you increase light, you can begin to bring these inner leaves up this curve, hopefully into this plateau area, so that now the majority of leaves on your bush are photosynthesizing at their maximum rate. And that will increase the amount of carbohydrates that you can move into your reserve pools. So pruning is very important in terms of light distribution into the interior of your plant canopy. So I think most of you know, or all of you know, about pruning. Southern highbush and northern highbush are pruned a little bit differently, at least in the States they are. Southern highbush, we always summer prune. And the reason for that is because we have very long growing. Our fruit comes off the plant in early May. We have a very long growing season after that. And so we go in and we top the plant at about 1 meter after fruit harvest. That allows sufficient time for new shoots to grow and then new flower buds to differentiate down those shoots during the fall. We will also go in and do some shoot tipping after we do that heavy pruning. For northern hybrids, typically they're winter pruned. Cane renewal is done. We'll take out maybe a third of the older canes, and then you'll remove all the twiggy weak growth, as we do in southern highbush. The other thing that's done actually in both of these types of blueberries is that if you have a cultivar that's a very heavy cropper, you will go in during the winter and do cutbacks on those shoots simply to remove some of those flower buds, so you don't get into that situation where you have an excess number of flower buds. How else can you increase light into the canopy? Well, there's been some recent work done where they've looked at reflective mulches, silver and white, and what they find is that those mulches reflect light from the ground back up into the canopy. And when they do that, they're able to get some yield increases. Now, this is recent work. It's only been done with one northern highbush cultivar. It's not commercially done in the States at all. So I don't really know how effective this is going to be, but at least it's being looked at in terms of increasing the photosynthetic rate of the entire plant canopy. So maintain healthy leaves after fruit harvest, and prune and perhaps train to maximize light exposure to the entire canopy will help reserve accumulation in the canes and the roots and help alleviate this problem of competition during bloom and leaf bud break.
Para não demorarmos muito, eu penso que agora esta parte é bastante clara, né? Vocês entenderam que com tudo isto, nós na altura da poda é que nós vamos equilibrar a planta. Portanto, o momento da poda é extremamente importante. Uma das coisas que era importante, nós tivemos a ver, a fotossíntese é muito baixa, estamos limitados por fotossíntese, a fotossíntese faz a um nível de radiação muito baixo. O que acontece? Ela explicou, atenção, mas nós, o que nos interessa é todas as folhas da planta que contribuem para esta acumulação de reservas. E então, o abrir muito a planta, não deixar que fique muito ensombradas as folhas, né? É uma das formas que nós temos para melhorar a acumulação de reservas, para que depois, portanto, atenção a tudo aquilo que nós temos que fazer. Portanto, aqui o que é preciso chamar bem a atenção é exatamente a nossa planta tem que estar bem aberta e temos que estar com uma, enfim, chamamos ocupado, não é? Ocupado muito, com uma radiação propícia a toda essa fotossíntese. A história dos ecrãs refletores já foi utilizada noutras culturas, portanto é uma coisa que não é nova, está a ser usada no mirtilo e o resultado é igual às outras culturas. Portanto, nós quando pomos ecrãs refletores, o que acontece é que estamos a aumentar a capacidade fotossintética das plantas e, portanto, estamos a aumentar estas nossas reservas da planta. E por isso ela tem aqui algumas das, e vai explicar mais, algumas das razões por que deve fazer.
And finally, it's important when you have cultivars that produce too many flower buds. Pruning is also important because it can help decrease the flower bud number. And if you decrease the flower bud number, you can help promote vegetative bud break the following spring. So again, this is work we did with southern highbush, where we looked at flower buds per centimeter cane versus vegetative bud break. The greater the number of flower buds, the lower the number of vegetative buds that broke. And that's a problem because that's going to be your canopy and supply your current photosynthate. And that translates, this is the same work, that translates into a severe reduction in canopy leaf area as flower bud density increases. So the more flower buds you have, the less vegetative bud break you're going to get in the spring, the less leaf area you're going to have during the course of fruit development. And that's going to translate into a decrease in whole canopy photosynthetic rate, which is what we measured in this same experiment. So as flower bud number increases, we get a decrease in vegetative growth, and we get a decrease in whole canopy photosynthetic rate. And that brings us back from this being the major limitation to blueberry fruit yield, and now we've added this limitation to blueberry fruit yield because we've reduced the amount of vegetative canopy that supplies current carbohydrate to support stage 3 of blueberry fruit development. How can you increase vegetative bud break after all, you've done everything you can for reserve accumulation? How do you increase vegetative bud break? Well, experimentally or perhaps in a tunnel, you could use CO2 enrichment. You can't really do that commercially very well, but we are able to increase vegetative bud break when we use high levels of CO2. Probably more importantly is you need to choose cultivars where you get sufficient chilling for both floral and vegetative buds. And so what we have here, so vegetative buds are in blue, floral buds are in red. This is time of chilling along the x-axis in both cases. This is days to 50% bud break, and this is the maximum percent of buds that break. And the bottom line here is that for a given cultivar, the more chilling it gets up to its chilling requirement, the faster bud break is going to be, and the more buds are going to break. And maybe more importantly is that vegetative buds have a longer chilling requirement than floral buds. And so you have to be in an area and pick a cultivar where you're going to have sufficient chilling for your vegetative buds. Your floral bud, if you have sufficient chilling for your vegetative buds, your floral buds are going to be fine because they're beyond their chilling requirement at that point. So it's the vegetative buds that are really critical. Another thing that we do that I realize you're not able to do is we're able to use chemicals, primarily Dormex, to promote vegetative bud break in cases where we don't have sufficient bud break occurring. So these are data from our plots. We're looking at Dormex at different concentrations and looking at leaf area. We can see a significant increase in leaf area when we use Dormex. We apply Dormex in the dormant season, usually right at the end of December. And then that, because we have increased leaf area, we get greater fruit weight because it's supplying current carbohydrates to the fruit. This is what it looks like. And so here's a row of southern highbush. This side was sprayed with Dormex. This side was not. And you can see the big difference in leaf bud break. And so all of our growers in Florida use Dormex every year in order to get uniform vegetative bud break so it can supply the current photosynthesis required for the developing fruit. And then that just translates into an increase in yield. And finally, what you can do is you can, as we've already talked about, particularly for heavy flower bud cultivars, you can decrease the number of flower buds. For any cultivar, we recommend for our growers that they do not allow it to fruit the first year, and in some cases, do not allow it to fruit for the first 2 years. This is some work that was done, again, out of Bernadine Strick's lab, where they looked at different cultivars of northern highbush blueberry. This is a picture of Duke. They did not allow these cultivars to fruit the first 2 years, and then they allowed them to fruit the third year. And so after 2 years, this is what Duke and all the others look like. You can see a reduction in the top in terms of growth, but here's the critical thing. Look at the reduction in root growth. So if they allowed it to crop, they got a huge reduction in root growth compared to when they removed the flower buds for the first 2 years. After the third year, that translated into an increase in yield. Again, cultivar differences, Elliott responded very, very well to that. And so there was a tremendous increase in yield in the third year in the plants that were not allowed to crop the first 2 years. So if you look at it in the short term, okay, you've lost 2 early years of production, maybe. But if you look at it in the long term, where you're maybe growing this plantation for 7, 10, 15 years, in the long term, you've made up for that by a lot because you have a much healthier plant.
Bom, penso que aqui também é suficientemente claro. A única coisa que vos vou chamar a atenção, aquilo que foi dito anteriormente, é tenham em atenção que quanto mais flores têm numa planta, menor o crescimento vegetativo. Portanto, a competição dá-se, tipo, vocês têm muitas flores, deixam muita carga, digamos assim. Vocês vão deixando da carga, normalmente o agricultor, pois a carga é o que me interessa, não é? Portanto, os frutos, e vai deixando muita carga. E o que é que acontece? Vai acontecer de que há competição e vamos ficar com menos folhas. Ao ficar com menos folhas, o crescimento vegetativo vai ser reduzido. Não tenho mais flores, tenho menos crescimento vegetativo. E este balanço, se nós continuarmos sempre neste ritmo, chegamos a uma certa altura e dizemos, os meus mirtilos não estão tão feios porquê? Não é? Portanto, este equilíbrio da planta é crítico. E foi o que ela vos explicou. É evidente que acrescentar CO2 não vamos fazer, enfim, nós aqui no Norte está toda a gente em ar livre. Não. Outra coisa muito importante é a atenção que a necessidade de horas de frio para os gomos florais e os gomos vegetativos não é igual. É sempre superior nos gomos vegetativos, portanto acontece muitas vezes, nós, enfim, pondo mirtilos do norte no sul, o que nós vemos é que damos as flores, mas depois aquilo não dá, ficamos sem folhas, ficamos sem parte vegetativa. Isto até dá frutos, mas depois não sei. Bom, Dormex é proibido em Portugal, portanto Dormex está fora de questão, é só dar-vos a conhecer que estas coisas existem, e este último diapositivo fala por si próprio, não é? Pronto. Isto é o que se diz sempre, atenção, não começar a colher o mirtilo demasiado cedo quando nós estamos em instalação da cultura, não é? Pronto, ok. Obrigado.
And so just to summarize what we've talked about today, Pedro and I have talked about today, carbohydrates, I didn't talk about nitrogen, I'm not going to, but carbohydrates are translocated from source to sink. The sinks compete with each other for the available source. These sources and sinks can and do change throughout the season. And we've identified 3 source-limited periods for blueberry fruit development. The greatest limitation appears to be due to competition between the flower buds or young fruit and the early leaf bud break, and this is competition for the reserve carbohydrates that were accumulated the previous year. And I'll leave you with the slide you've already seen that indicates at various times of the season the sources and the sinks on the blueberry plant. And with that, I'm done. Okay, thank you.
Eu peço à organização que me diga, estamos atrasados, não é? Estamos muito atrasados. Portanto, eu peço desculpas, não há perguntas nem respostas, portanto, a Rebeca vai ficar connosco amanhã que estará connosco, portanto, tem a oportunidade para, enfim, se abordarem dela e tirarem algumas questões. A Rebeca, como não fala português, terá que ser em inglês e, portanto, mas terão a oportunidade de fazer as perguntas. Peço desculpa, mas estamos muito atrasados. Muito obrigado.